talk about a little bit about very important cells, which is called the myeloid-derived suppressor cells, and its impact on the crosstalk between tumor and immune cells. The research interest of my group is focused on four areas. One of the cancer escape from immune response, cancer immunotherapy. The third one, identifying the molecular signature of cancer and immune cells. And the last one, which is of our ongoing interest, is correlating cancer stem cells to clinical and immunological performance. Most of the data presented in this talk is funded by two grants from Science and Technology Development Fund in Egypt. The talk agenda is focusing on 5 points. One is immune cell dysregulation in cancer, emergence of MDSC, which is myeloid-derived suppressor cells in preclinical mouse model, emergence of MDSC in human solid and liquid cancer. I'll present some of our data, subsets of MDSC based on our recent publication, and the conclusion. Before I start, actually, the interaction between immune cells and the cancer cells is like a tag of war game. It's evasion and escape mechanism, and it is a matter of who is going to be stronger and intelligence. And this escape mechanism can be categorized into intrinsic and extrinsic mechanism. In case of intrinsic mechanism, it includes cellular and molecular. The cellular one is include circulating tumor cells, cancer stem cells, autophagy, and the other cellular mechanism. The molecular one includes antigen editing of cancer cells, down regulation of expression of class 1 and the class 2 MHC, expression of PD1, PD-L1, and the other tumorigenic factors. In case of extrinsic mechanism, in particular immune cells, the cellular arm include emergence of 3 regulatory cells, MDSC, Treg cells, and the subsets of NK regulatory cells. The molecular one is still also the hot topic is PD1, PD-L1 expression plus suppressive mediators. There is flexibility of tumors of immune cells in response to tumor microenvironment, and we can roughly divide cells, immune cells, into type 1 and the type 2. Type 1 usually is with good reputation in immune response, but type 2 like DC2, dendritic cells type 2, macrophage type 2, neutrophil type 2, Th2 type 2, and the Treg cells, all such kind of type 2 cells lead to tumor progression, unfortunately, because they produce pro-angiogenic factors. They enhance tissue remodeling and enhance immune suppression. Two of the important cells in extrinsic mechanism is and share the immune evasion is myeloid-derived suppressor cells and regulatory T cells. These are actually increase in numbers under the impact of tumor cells. And these cells plus the TGF-beta IL-10 produced by tumor cells, regulatory cells, and other mediators by MDSC, they induce shutdown of T cells. I'll focus on MDSC, although I'm also my group interested in Treg cells, and we published a couple of papers. MDSC is a population of early myeloid cells from bone marrow, are expanded under various settings like cancer, chronic inflammation, and they are potent inhibitors of anti-tumor immunity. And this, of course, results in progression of and relapse of tumor. MDSC, based on our results and the other groups in the middle here, they produce several factors which induce type 2 response, like IL-10. They block dendritic cells, they block regulatory T cells by production of IL-10, arginase, TGF-beta. They also produce plus arginase, NOS, ROS, and cysteine, which unfortunately shut down the activity of CD8 cells, as well as CD4 cells, macrophage, and the natural killer cells. So these cells actually play very important role in mediating the suppressor function of a tumor microenvironment. When I looked yesterday at the number of publication, you cannot imagine here is MDSC, the total number of cells is over 61,000, and in case of cancer, 56. So most of the publication actually is linked to cancer. And in Egypt, I found appreciated number of publication, but it includes from inside and outside Egypt. Actually, my story with MDC started when I was in US in like 15 or 17 years ago. I worked there as assistant professor at Medical University of South Carolina for 10 years. At that time, I was submitting RO1 grant, and I found that I used cyclophosphamide to precondition the host to enhance CD8 T cell response. During that time, I found a very interesting observation, which is expansion, kinetic expansion of cells in blood and other organs like spleen, bone marrow. And when I looked by flow cytometry at these cells, we found them they are myeloid-derived suppressor cell. At that time, it was immature myeloid cells, but later on, it's named as MDSC, and was associated with splenomegaly. And it started by day 5, and then peaked by day 12, and then sustained until day 27. When we looked at these cells at different time points in different lymphoid compartments, we found them everywhere, even in peritoneal exudate. When we looked at the relative and absolute number of these cells, we found them, they are, and the kinetic, we found them, they peak, as I mentioned to you, at the absolute number at day 6, and then sustained at later time points. We asked actually what these cells are doing there, and we come into the conclusion that we published in a couple of papers, and the last one actually was with the conclusion that these cells emerge from bone marrow to see different lymphoid compartments in the periphery with very potent immunosuppressive function. So we moved in from preclinical to clinical studies. I started this in 2007 when I was in US, and then when I came back in 2010, I worked on this in different couple of cancer types. We started with different screens, these cells in different cancer types, including breast, lung, colon, and other solid tumors. And we found that when we compared the number of these cells in normal volunteers were very low. I'm talking about the percentage and absolute number. And we identified these cells as Lin negative, CD33 positive, CD11P positive, HLA-DR negative. When we looked at the different stages of breast cancer, we found the number increased with the cancer stage, from stage 1 to stage 4. When we looked at the limited cancer and extensive cancer, I mean with metastasis, we found also correlation between more increase in the number in extensive as compared to limited at the percentage and the absolute number of these cells. And this one representative scan of stage 4 cancer patients, we found the high number of these cells, which reaches sometimes to 26% in a patient with melanoma. And in most cases, the mass of tumor was large. Then we asked whether the chemotherapy can eradicate or increase or stabilize the number of these cells. We tested this question in breast cancer patient treated with ddAC protocol, followed by ddT. ddAC is doxorubicin cyclophosphamide, and then later on with paclitaxel. And we counted the cells in normal stage 4 and then with ddAC, and we found that still the cells still there in normal 1.1, the best scenario, and then 22% the worst scenario in breast cancer patient, and after chemotherapy still there. And then we looked at compared the DDC stage course and the DDT, and we found they peak at the end of DD, and then after that they actually decrease in number. So the conclusion was that increased the MDSC level during ddAC, but not during DDT therapy. And then when asked if these cells actually expand or emerge in other types of cancer, and we were interested in hepatocellular carcinoma, and then but before we asked if they can be also expanded in HCV patients. When this is the representative flow cytometry from different groups, and this is from control, naive, responder, and the non-responder. Interestingly, we found a non-responder, they have more cells than a responder. And of course, a naive HCV, they don't have much cells. And when we compared even in responders post-treatment, the cells as compared to a non-responder, and the responder, we found the cells actually decreased in number in sustained responders. We looked at the expression of IDO, NOS, and the CD3 zeta, which are the marker for the suppressor function of MDSC, and we compared in between responder and the non-responder, and we found actually, regardless, the MDSC express these three molecules in especially IDO, even in naive HCV. But interestingly, we found in non-responder high level of CD3 zeta, which is one of the hallmark of the biomarker of this suppressor function of these cells. And then we moved to HCC patient and compared to cirrhotic as control, and we found a high number of these cells in HCC as well as cirrhosis. And we looked at the blood, compared the blood and acidic fluid. We found acidic fluid doesn't have much of these cells. Most of cells are in circulation and blood of HCC patient. And the number of these cells in HCC patient was correlated with the high level of alpha-fetal protein. Then we looked at the function of these cells. We looked at cell proliferation and cytokine production, mainly IL-2 and the interferon gamma, and correlated this three function with the number of cells in blood. When we have high number of cells, like 22%, 26%, 24%, the cell proliferation decreased very much as compared to when the cells are low in patient with low number. This is the same thing with IL-2 and the interferon gamma. Then we sorted these cells from these data from unfractionated peripheral blood monocyte. But when we sorted these cells and looked at the and normalized the number and co-cultured these cells with T cells and activated this with CD3, we found when we have high number of high number of cells, they suppress the T cell completely. I was working with a clinician, and he was very oncologist, interesting in the kinetic of these cells after, at that time, treatment with gozerlin and the other aromatase inhibitor. And we found this kind of treatment, the number of cells dropped and almost eradicated from the peripheral blood after this kind of treatment, which opened a new window for our study. Then we asked if these cells, MDSC, can be categorized into monocytic and granulocytic, and you can determine either of them using CD14 and CD15. Monocytic are positive for 14, granulocytic for CD15. We started this actually just recently, like 3 years ago, and at our Center of Excellence in Cancer Research at Atlanta University, and we found most of cells actually are monocytic rather than granulocytic. And the monocytic, by the way, are more suppressive than granulocytic subtype. And we looked at these cells in monocytic and granulocytic after gating on MDSC, and we looked at the percentage and the absolute number with in breast cancer patient before chemotherapy and after chemotherapy. We found a high number of cells before therapy in terms of monocytic and granulocytic. However, after therapy, we see more monocytic, which is, of course, problematic. And we looked at ALL patient, acute lymphoblastic leukemia patient. We looked at the cells. The total number of MDSC was very high in peripheral blood of these kids. And we looked at either before, during, and after induction of chemotherapy. And we expected low number after therapy. However, we found still high number of cells, even at the level of percentage and absolute number of these kids. We looked at also monocytic and granulocytic, and we found high number of monocytic than granulocytic, which was consistent also the same, because we are interested now in monocytic. We found the same in observation on colon cancer and other lung cancer, lymphoma, ovarian cancer, head and neck. Because of the time limitation, I showed you some only ALL and breast cancer and lung cancer. And actually, in this one, we started to look at the molecular signature of MDSC in ALL patient. What we did here, we prepared the RNA from blood of ALL patients and run microarray for microRNA. And we found a very interesting observation here. We found some dysregulation, a very actually interesting dysregulation of certain microRNA in before as compared to during and after chemotherapy. We then focused on, we did actually a lot of bioinformatics data on innate immune cells, adaptive immune cells, including other cells, but I'll show you only some of the data on MDSC. What we found actually about 13 microRNA upregulated, downregulated when we compared patient before versus control, and 19 up, 18 down when compared patient during therapy versus control, and 8 and 16 down when we compared the patient after therapy to control. But we asked whether is there MDSC share certain microRNA regardless chemotherapy, and I guess, I'm not sure if I have it here. Yes, this one. We found 13 microRNA, which are this one, shared regardless the treatment before, during, and after. We found 13 microRNA are shared. And we confirmed the expression of these microRNA, 13 microRNA, by real-time PCR, and we found very interesting results concerning with certain microRNA. And we looked at this specific microRNA, the pathway, and we found they are linked with RAS, PI3K, mTOR, and these actually are very important based on literature. They are very important pathway in mediation of immunosuppressive function of MDSC. So the conclusion is that both solid and liquid tumors show direct correlation between high level of circulating MDC and the clinical stage. Those dense AC but not those dense paclitaxel associate with number of MDC. And by the way, we found the other chemotherapy also induce emergence or expansion of MDC, especially gemcitabine, but I didn't show here. Both tumor and ddAC induced MDC are immunosuppressive, as I showed you. Emergence of MDC predicts tumor genic and immunosuppressive tumor microenvironment. And now we are actually working on standardization of MDC analysis as a biomarker. And with this one, I'm contributing to consortium between two labs in Italy, one from Spain, Germany, and I believe another one from Spain, and my lab in Egypt, to standardize MDC analysis protocol by flow cytometry to be used as a biomarker in different cancer setting, either solid or tumor. And we just started this. It's funded from 6 months ago. We just started this very interesting project, a multi-center project. We published our some of the data I showed you in couple of 7 or more publication. The most recent one is myeloid derived suppressor cells and regulatory T cells share common pathway related to microRNAs that are dysregulated by ALL. This was published a couple of months ago in Human Immunology, and this was very interesting paper based on bioinformatics and the lab data using microarray and real-time PCR. And based on this one, we are going to actually focus on precise microRNA to be used hopefully as a biomarker. As I mentioned to you, current ongoing studies, the standardization of MDSC analysis as a biomarker through consortium projects funded by Academy of Scientific Research and European Union. And we are thinking of how to target MDSC, either at the level of myelobiosis from bone marrow, or inhibition of MDSC activities, or depletion of MDSC, or using some of common, multiple pathways at the same time. At the end, I would like to thank my colleagues at Medical University of South Carolina who started this project with me, and my colleagues at Tanta University and other institutes. And just yesterday, one of my students, a PhD student, just got her PhD, and her work was focusing on MDSC and the cellular and the molecular signature, especially the role of microRNA. And she is Mona Zidan. She is working at 57 Children's Hospital at Cairo. And this is some of my team at Center of Excellence of Cancer Research at Tanta University, which I established after I came back from US in 2010. And from a grant from Science and Technology Development Fund. And this is the website for the center, and is working very nicely. And we are not just doing research, we are training next generation scientists.